It may not come as a surprise that a cell's main goal, like all living things, is to grow and reproduce. Unlike you and me, however, our cells undergo a cyclic process of growth and division over their lifetimes called the cell cycle. The main phases of a typical cell cycle include the G1 phase, S phase, G2 phase, and M phase. All the phases are essential for the cell to function properly, so let's take a closer look at each one. The G1 phase is commonly referred to as the growth phase because this is when the cell is growing and monitoring its environment in order to determine whether or not it will continue with the cycle to produce more cells. This is similar to how humans grow and develop over the course of our childhood and adolescence. In this phase, the cell synthesizes messenger RNA and proteins that will eventually be prevalent during mitosis. During the S phase, the cell begins the process of replicating its DNA, creating a second copy of it so that both daughter cells can have a full set of chromosomes. G2 is the period after a cell has fully replicated its DNA. This phase is where the cell makes sure its growth and DNA replication has been successful and does all of the necessary repairs. This is the final preparation before mitosis. But how does a cell know when it's time to transition from one phase to the next? A group of researchers looking at cell growth in mice set out to explore some of the traditional knowledge on the subject. Cells know when to move on in the cell cycle in part due to the contribution of special proteins called cyclin-dependent kinases, or CDKs. CDKs are enzymes that direct reactions and structural changes that help a cell transition between the phases of a cell cycle. However, only when the right cyclins can pair with the right CDKs does a cell move from one phase to the next. In their study, the research team focused on 4 specific classes of cyclins called A, B, D, and E. They found that different cyclins were responsible for various key processes in the cell cycle, and that their removal could halt the cell in its tracks, preventing it from eventually dividing. Assuming these cyclin checkpoints didn't go wrong at any point, the cell can successfully accomplish division, which leads us to the M phase. In this phase, the cell actually undergoes division. This division can occur via mitosis or meiosis, but here we're just going to focus on mitosis. In the first stage of mitosis, known as prophase, DNA is condensed into double rod-shaped structures called chromosomes. Since the S phase replicated all of the DNA within the nucleus, we now have 2 identical copies of each chromosome. These copies, known as sister chromatids, are attached to each other at a point called the centromere. At this time, the nuclear envelope also disintegrates. In the meantime, centrosomes start to organize microtubules from opposite sides of the nucleus and bridge of spindle fibers forms. In metaphase, the chromosomes are aligned at the middle of the cell. Microtubules from both centrosomes have attached to the chromosomes, linking the sister chromatids to the opposite poles of the spindle. In anaphase, the centrosomes start tugging at the line of chromosomes, pulling the sister chromatids apart. During telophase, the chromatids arrive at their final destinations on opposite sides of the cell, where they begin to unravel back into the uncondensed form of DNA that usually takes place in the nucleus. New nuclear membranes also begin to form around the individual sets of DNA. Cytokinesis is where the actual division and cell division happens. Once the chromatids are separated, the cell membrane begins to pinch closed around the center of the cell, splitting it in two. 2 distinct daughter cells that are genetically identical to the original are created. Now the cell is reproduced, sort of like a mother having children, except instead of creating a genetically distinct child, it would be like if your mother had split into 2 and become 2 smaller versions of herself. It's because of this reason that we should probably be glad we don't replicate like our cells do.